Now, in this last session of the lectures or the series of the lectures on the eyeball, we are going to discuss here the third component, the inner coat of the eyeball, that is called the nervous coat, inner nervous coat, or you can say its name is the retina. Now, before understanding the retina, have a look at this in this picture that we have been actually discussing for last two classes here. In this plan, we have actually divided the eyeball into six parts here, and in the outer fibrous coat, most anterior 1/6 it was the cornea, and the later you see all four, I say 5, 6 they are sclera. And then you are actually getting here into another, this is red color, that is in fact here posterior 4/6 is the choroid, this is the middle vascular pigmented coat. Posterior 4/6 is the choroid, that is underlying the 4/6 of the sclera, 4/6 of the sclera. Sclera has 5 parts, but we said the 4/6 of the sclera, this is the hidden sclera. Under that is this vascular pigmented coats component that is called choroid here. And then when choroid finishes here, next component of the vascular pigmented or the uveal tract start, that is the ciliary body. And ciliary body in fact is underlying the visible part of the sclera that you are actually seeing here. Uveal tract is not yet finished here because 1/6 more is required here, and this 1/6 more means it is the continuation of the ciliary body, and this time its name is the iris. So, the iris will be under the most anterior 1/6 of the fibrous coat, that is the cornea. So, under the cornea is under the transparent cornea is the iris, and in the center of the iris there is an opening that is called the pupil. Now, after this, you know that this is a ciliary body, and ciliary body has the suspensory ligaments or the zonular fibers which is actually holding the lens capsule, that is the elastic capsule here. And if you are actually passing here a line that is actually touching here the middle of the lens or the posterior aspect of the lens here, this line is again important because anterior to this line is the anterior 2/6 of the circumference of eyeball, means eyeball itself with the 2/6 means almost 1/3 is actually anterior to this line, that is visible. So, under the visible are the apparently which you can see the part of eyeball, it has the cornea and 1/6 sclera. Under that, under the cornea is iris, and under the visible sclera will be the ciliary body, and surely in the middle of the ciliary body will be the suspensory ligaments or the zonular fibers which are actually catching hold of the lens here. So, that means these are in fact they comprised of 2/6 of the eyeball, remaining 4/6 is hidden. So, if for example you see in this hidden portion we have got the 4/6 hidden the sclera, and then will be the choroid, this is 4/6 also, and then will be the nervous coat that is called the retina. This indicates the retina does not occupy the entire all 6 parts of the ball of the eye or the eyeball here. It will occupy only posterior 4/6, means the retina will be related to the posterior segment of or say posterior compartment of the eye, in such a way that if this line which we have actually divided in such a way that it has divided the eyeball into the anterior 2/6 and the posterior 4/6. So, that means in the posterior 4/6 will be related your cornea, I say related your retina, not in the anterior 2/6. Anterior 2/6 you know that this is called the anterior compartment. So, retina basically is a game player, the real game player is this nervous coat here, where all these things, the rays whatever actually have reached there, the stimuli have reached there, transduction will take place, its transmission onward from first order neuron to second order neuron into the tract even. This is the work of the retina, which occupies here the posterior compartment of the eyeball. In the posterior 4/6, this is those 4/6 which are not visible here in the circumference of the eyeball from the front, means all that is hidden within the orbit and the orbital fate. Actually, retina lives there. Now, as far as the retina is concerned, actually retina is the last thing that we are actually discussing, but in development, when the developmental point of view, retina basically is the first thing that develops. In fact, it is the gift of the brain to the eyeball structures, because you see that in such a way. So, you can say like this is brain, you can say here, okay, this is brain, forebrain. From the forebrain, there will be an extension here like a stalk here. This is called the optic stalk. Optic stalk terminally here, at the most distal portion, it get bulges here like this. You call it optic vesicle. But this optic vesicle actually will remain not like a vesicular here, it will in fact be compressed from the front, so that it will adopt or assume a shape of a cup. Now you can say, you can call it now optic cup. You see, it will not remain like a vesicle like this, because this optic vesicle will have actually two layers. You can say it will compress in such a way it will collapse. It will not remain like a vesicle, it will become now collapsed ball now. Like this. This is called the optic cup. So you can imagine that the vesicle which was single layered, the cup will be now double layered. In such a way that this anterior, or you can say inner layer is this is its inner layer, and this will be its outer layer. These are the two layers, and in between there is a space, and that space is called the intraretinal space. This is the development of the retina. And you see, this development of the retina is there, but now this area means this inner layer which I am actually showing you here, this inner layer is there and this outer layer is there. Outer layer and inner layer together they will constitute 10 layers. In such a way that this outer layer which I am actually showing you here, this outer layer basically it is forming its layer number 1, that is called the retinal pigment epithelium. And remaining layer which I am actually showing you, it is not pigmented, this is actually called the neuronal layer, sensory layer you can say. And that neuronal layer will in fact will give rise to all other layers you can say. Next means 9 layers it will form, from 2 to 10 you can say. All the layers will be constituted, will be actually developing from this sensory layer, and at a spot or at a position, you see it was like a vesicle here, it was like vesicle. Then it collapsed here, collapsed at a spot, you see at a spot where it collapsed here in such a way that this double circle which I am actually showing you here like a cup here double, that will occupy posterior 4/6 of the eyeball, means it will be under the choroid. This indicates that this retina which I am showing you all 10 layers, out of these 10 layers the outer layer will form the one layer, that is called the single layer, short columnar cells or cuboidal cells. These are called the retinal pigment epithelium. But the remaining layer that will be actually present here, that is called the neuronal layer, or you can say the sensory layer, that will in fact give rise to next 9 layers you can say, including rods and cones, the first order neurons, the second order neurons, the interneurons in between them, all these cells are actually being constituted by this inner layer. And in between there is a space, intraretinal space, but later on this intraretinal space in fact become no longer, and both these layers, layer number 1 that is called the outer layer, that will give rise to layer number 1, and the next will be 9 layers, means actually in contact will be the second layer and the first layer. First layer already there, and second of the 9 layers, means from 2 to 10, the second of these 9 layers that will actually fuse with, and they will be actually in touch with the first layer. Means in the second layer what is present? The outer segments of the rods and cones are there present, they will be actually be invaginating, they will be embedded now partially into the pigmented epithelium. So intraretinal space will be no longer. Intraretinal space is no longer, but there is a cup-like space is present here. So in the space, in the space means in the vacuum in front of the cup, that is present. That is present means this space, and where the bending takes place, like here this bending means where your sensory layer, you can say the area where my photosensitive layer become continuous with the non-sensitive, non-photosensitive layer, you can say. This area is called the ora serrata, means this is the area. So this I am actually showing you a double layer. Interiorly will be from 10 to 9, I say from 10, 9 up to 2, and then this outer layer will be my first layer. In between there is no space now, all 10 layers are in series. But now the retina is not like a say optic vesicle, it is like an optic cup, like an optic cup now, and cup is there, so this is the cup now. But the cup is made up of 10 layers, the wall is made up of 10 layers, but these 10 layers have actually been developing from the 2 layers here, and these 2 layers actually be bending at this spot, and this spot is called ora serrata. Serrated means it is having pointed here digitations here. This is called the ora serrata. Ora serrata is actually the spot where there is a sudden transition of the photosensitive layer into the pigment layer. You see, this is here is a bending here. Means this is the bending between, you say, the bending of the 2 parts of the retina. You can say interiorly, say inner one, this is the sensory, and outer one, that is the pigmented here, and there will be actually a digitations here, indentations here, you call them ora serrata. This is called the ora serrata, and ora serrata will be present here. Being ora serrata is in fact is the limitation. Beyond that will be the anterior compartment of the eye, so that means, and behind that will be the posterior compartment of eye, means behind will be the forceps, means retina will be totally confined here to the posterior forceps in such a way that because it is a cup. So that means if it is a cup is there, so there is a gap now, there is a big vacuum, there is a empty space here, that empty space will be filled by a transparent jelly like or the gel like substance here, that is called the vitreous. So that will be the vitreous now. Here now I am actually filling here the fluid, that is called the vitreous. It is actually kind of a body, it's called the vitreous body also, it is kind of a combined gel compartment. So it is like a gel in such a way, that means now you can say inside you have got here the vitreous. So that means your retina is actually compressed between vitreous in front, I say vitreous inside, and the choroid outside. So you can see this green color retina at ora serrata, you see that is here. So it can't actually go beyond the ora serrata. Here this is a gel like substance, it is called the vitreous here. Means it is attached here, means attached to what? Means outside it is attached here to the choroid. And when it is attached to the choroid, choroid is filled with the choriocapillaries. So the retina outer portions, you see almost 1/3 of your retina outside here will be in fact getting its nutrition from the choriocapillaries, highly fenestrated choriocapillaries. Means your retinal pigmented epithelium, even your rods and cones, outer segments, etc., they will actually get the supply or nutrition from the choriocapillaries. But the inner sensory portions here, neuronal layers and all these things in fact will get the supply from central artery of retina, and central artery of retina, you know, that is passing at the middle of the optic nerve, you see that, and it enters into the retina here. This is called the central artery of retina, and that spreads here into 2 branches, upper and lower, and each one of them will divide into the nasal and the temporal branches here. And these 2 branches which I am actually showing you here, the 2 branches of your central artery of retina, very important branch of the ophthalmic, and ophthalmic is the branch of the internal carotid, and that branch is in fact entering here and entering and spreading here, and these 2 are the end arteries, because these 2 branches in fact will not anastomose with each other, and these are the end arteries. So if for example you are watching in this, so this retina which I am actually showing you here, this is outside having a choroid, because choroid is also available in the forceps, retina is also available in forceps outside here, and inside has the vitreous now. And vitreous is there, means vitreous is not just giving the globular shape to the eyeball, but is actually pressing the retina from inside here, so that the 2 layers of the retina, you see the sensory and the pigmented layer, which have fused here, intraretinal space is no longer, their fusion and their approximation, or we see their fusion or their contact with each other will remain as long as the vitreous stability is there. When there is a vitreous degeneration, you see pressure will be less now on the 2 layers, and these 2 layers will start separating. This is called retinal detachment. And with the 2 layers will start separating, means which 2 layers are really separating? Actually first and second layers are separating, and the second layer is lying here, the outer segments of the rods and cones which have been embedded in the in their tips are actually embedded in the retinal pigment epithelium here. So they are actually coming out of them. It is coming out of them and getting away from them means their nutrition is interrupted here, because they are getting their nutrition from the pigment epithelium. Actually, pigment epithelium is not giving them nutrition. Outside the pigment epithelium is lying the vitreous, and vitreous is having choriocapillaries, and choriocapillaries are highly fenestrated. So there is a diffusion of gases is very easy there. So rods and cones in fact will be deprived of their oxygen supply, and they will undergo degeneration here, and this will lead here to the blindness. So this is, you are actually watching here in this sense. So ora serrata is that spot where the photosensitive part finishes, and transition will be to the simple non-photosensitive part here. This is actually, and this is embryologically, this is an area, you see, this is the area where there is a bending of your inner layer and the outer layer, the neuronal layer and the non-neuronal layer, or the pigment layer. And here you can say, and neural layer will give rise to 9 layers from 2 to 10. But the first layer already available, that is actually the retinal pigment epithelium, which is already present here. Now, in this especially, I told you there is attachment you have seen, and vitreous, which I am actually showing you, is giving not just the globular shape to the eyeball, but at the same time it is a transparent, and it has got its refractive index, and you see that it has got its refractory power. So it is a medium of refraction also, a transparent gel-like substance here, and it has been actually given by nature to the human being only once. Once there is actually damage to the vitreous, will not undergo now reformation or regeneration here at all. It is formed once and for all. You see that it is formed only once. And here, if for example you are watching in this, so you have one thing, 2 basic layers of the retina, which in fact pigment layer and the neuronal layer, and neuronal or sensory layer will give rise to the next 9 layers after 2 up to, you see, after 1 from 2 to 10 you can say. And ora serrata, already I have told you that this is the area of the transition from neuronal to the epithelial layer. Now, if you will actually look at this, now some 5 to 7 here this is the middle of, or the center of the cornea. This is center of the cornea. You are passing the plane through the center of it, then through the center of the iris, you say center of the pupil, through the center of the lens. And when you actually go back here, here will be the posterior pole, some 5 to 6 centimeters medial to the posterior pole. There is a spot which in fact will give you idea that from here the optic nerve fibers are leaving now. As optic nerve fibers are leaving, and that spot will be, you say, hardly it will be 1 to, you say, 1.5 mm in diameter here. Means this is the spot here, and that spot is called the optic disc. Means this is the exit of the optic nerve fibers. And when it is actually here, so it will penetrate what? When from all the fibers in fact will accumulate here from all the sides, from all circumference of your retina. These fibers will accumulate here, this gate now. Because in the room you see that there is all around, there are the walls, but there is a gate also. All the people actually will leave that gate here. So all the fibers in fact will leave this gate here. From inside, if you are seeing here, so this gate here, so all the fibers are now leaving. And that area is called the optic disc. And this is the optic disc which you are actually seeing. It is hardly, you see that is 1 to 1.5 mm in diameter. And that you are actually watching from the optic nerve fibers are now leaving from the retina. If they are now leaving from the retina, you can say optic nerve has been actually created here, and optic nerve fibers you are actually watching, and in this especially, this is called the optic disc, 1.5 mm in diameter. So you can say optic disc represents the head of the optic nerve. From inside you see that is the head of the optic nerve. Before that is the optic nerve fibers, now it is the optic nerve. So the top of the optic nerve in fact is actually the optic disc here. And optic disc is lying where? Location already I have told you that it is lying some 5 to 6 centimeters, 5 to 6 millimeters on the medial to the midpoint, I say medial to the posterior pole, from where optic nerve is leaving now. Okay, this is true. But if for example you are watching in this, and you see through the optic nerve is entering the central artery of retina. So if you are actually sitting inside the vitreous, and you are looking at the optic disc, in the center of the optic disc will be entering now the central artery of retina. Actually, right at the middle point of your posterior pole, just opposite, means in the straight line that passes through the middle of the cornea, through the middle of the center of the pupil, through the center point of the lens, and then is the posterior posterior pole in the center here. Right at this place there is another very important area which is in fact is having less vascularity, look like actually pale color area, that is called the macula. Optic disc is lying 5 millimeters medial to this medial to this macula. If it is lying medial to this macula, so how you differentiate is optic disc or the macula? So macula basically itself is a larger area. Larger means it will be around, you can say 5 mm area, 5 mm pale area is there. But one thing is more, in the optic disc in the center there is a central artery of retina, in the macula there is nothing like this. And the central artery of retina is actually emerging and is splitting up into the upper and lower branches, and upper and lower will have temporal and the nasal branches here. Nothing is actually like here in the macula at all. So that differentiates actually central artery of retina, differentiates the macula lutea, we call it the macula lutea from the optic disc here. Optic disc, because inside there is a pressure, this is called the intraocular pressure, and intraocular pressure is putting pressure here from the inside of the optic disc here. And because optic disc is supported here by the choroid which is already perforated, and then by the sclera which is already supported, lamina cerebrosa, so that area will be weaker, and due to the weakness and due to the pressure here, it will be actually having a kind of a variable excavation will be there. This is called physiological cupping. A cup like, you see, the optic disc will be like a cup, you can say. A variable cup will be there. This is called the physiological cap, which is called the physiological cup. Physiological cupping will be there, and the bulging will take place if the intracranial pressure is more, because this time the pressure is from behind. And if you are actually watching here, so this is now, because in all retina here, from all sides where the retina is available, just like a cup here, all it is photosensitive, except at the area, like in a whole room there are bricks and concrete, etc. But where there is a door, you see bricks are deficient there. Same is the case here. Means from all sides you have got the photosensitive layer, you have got rods and cones, etc. Except at a spot where there is the optic disc. Means because all the things are actually going through that, means at that area we don't have any photosensitive receptors or anything will be there. So devoid of any photoreceptors. Means that spot or the face, you can say, optic disc actually represents the blind spot. Mean here the visual stimuli cannot be in fact transduced into impulses at all. This is called the physiologic blind spot. But if there is any other area which is a photosensitive, if any other area actually can't transduce the impulses as a stimulus into an impulse, then you will not call it a physiologic blind spots. That blind spot is a pathologic, we call them scotoma. Now in this especially I say central artery of retina and optic disc is important here. Now for example, if you are looking at through ophthalmoscope, you are actually, so you look here, this is now the optic disc. So you can say the disc and the whole surrounding area around the disc, that is in fact you can say that is seen under the ophthalmoscope, that is called the fundus. And its visualization through the ophthalmoscope is called the fundoscopy. Means through the pupil, you put the light here through the pupil and you will actually watch the optic disc and surrounding area also. And surrounding area means in surrounding area there will be macular lutea also. So you can see the macular lutea. And the center of the macular lutea, there is a pit now in the center, that is called fovea centralis. So especially you see macula, I have already told you, the 5 to 6 mm lateral to this, lateral to the optic disc. It is a shallow depression, you can say it is completely free of blood vessels. And this is called the yellowish spot, you see the yellowish say macula, you call the macula lutea. Macula, central artery of retina, already we have told you the central artery of retina, it is supplying the inside neuronal layers, everything here. But in the macula, macula is not supplied here by the central artery of retina. The macula is again, though it is the central part, you see, though it is inside here, neuronal parts, everything is there, but it is not supplied here by the central artery of retina, that is supplied by the choriocapillaries. Macula means the central artery of retina is actually blocked here, macula will be spared. This is called the macular sparing. So in the macula is there, I already told you that in the macula is almost 5 mm in diameter, but in the center around 1.5 mm in diameter in the center of the macula now. There is a depressed or a shallow pit is there. Now you can say, for example, if you are actually watching all these 10 layers here in the macula area, this is retina. So, all 10 layers you are actually making here in this retina. Right in the center of this retina, I say center of in the macula here, this total will be diameter will be 5 mm, but here in the center hardly 1 mm or 1.5. You see the areas will be, you see 10 layers will not be present from inside, almost you can say it will be like this. Means many layers in fact will compress here, they will disperse. So, almost 4 to 5 layers actually they disperse. So, this shallow spot or shallow pit here that you are watching here, that is called the fovea. Fovea means pit. In the center of the macula, that is called the fovea centralis. Overall macula can be 5 mm, but this will be hardly 1.5 mm. In the center, the shallow pit is there. This is called the fovea centralis. The fovea centralis is a shallow pit in the center of the macula. And you can see here they are having you see no blood vessels. Already macula is less vascular. So, no blood vessels are there. Even in that macula, I say in the fovea centralis, there will be no rods. It is a rodless area. It is only the cones. You see this only the cones here, highest concentration of the cones. It is not just the cones here, but there will be highest concentration and density of the cones will be there. So, this area will be the area of highest visual acuity, sharpness of the vision will be highest here. And if I am actually showing you here the cones here, each cone in fact will be synapsing with one neuron in front, you can say. In front means in the next deeper layers there will be one neuron only, means one bipolar cell will in fact be connecting and synapsing with one cone. One cone means which cone? The cone that is lying in the fovea centralis. If the fovea centralis has got 1000 cones, for example, they will employ 1000 bipolar neurons. This will give you idea how much sensitivity they will have. So, that means these are the cones here and is almost a 1:1 ratio you can say with the bipolar cells here. And this is the sharpest vision you can say and is lying where? It is lying, for example, in the same line, a straight line. Same straight line means in the middle of you see in the center of the cornea that passes through the center of the pupil, the center of the lens, it will strike the fovea centralis. Fovea centralis is actually the central spot on the posterior pole where there is the sharpest, I say the sharpest, I say highest acuity in the vision has there. Now, for example, that is why when you are actually fixing some gaze, for example, focusing something, three things will happen together. If you see focus something here, you see that it is not just accommodation takes place here. Accommodation means your ciliary muscles will contract. As ciliary muscles will contract, ciliary body will come closer from all sides. Means ciliary ring will get narrowed down. Suspensory ligaments which are attached to the ciliary processes, they will get lax now. Over the elastic capsule will become less traction. So, the bulging of the lens will take place, more curvature, more convergence power, and nearer object will actually fall, which was actually falling behind the retina. This time it will now, image will fall on the retina because convergence power is increasing, refractory power of the lens has been added now. If the lens is not working, then plus refraction plus now you can say convex lens means plus power you have to add now. So, this is what we call here accommodation, and accommodation which part of the lens has to be utilized here? More bulging part, and bulging part is in the central part, and the central part means the rays or the light should actually enter here into the center of the say in the center of the lens, and in the center of the lens it will be possible only when the pupil will constrict. Means right at this point when there will be accommodation, there will be meiosis also, pupillary constriction must. Both of these works are being done here by the parasympathetic nervous system through ciliary ganglia, through short ciliary nerves. And this time constrictor pupillae or sphincter pupillae that will also constrict. But because you are focusing now, means you are fixing the gaze now. So, this time retina's which portion should be used? Macula, and macula which portion? Fovea centralis. That is why your both the eyes in fact will be medially converging, so that the fovea centralis actually gets the maximum light. So, that means here will be convergence also. If you will actually look at your finger here, you see your both the eyes in fact will be moving, your medial recti medial rectus of the right and the left both will be actually moving in such a way that the cornea will be converging now. I say eyeballs will be converging, so both the corneas will look medially. This is called the convergence. That is why in accommodation always there will be convergence also. So, another landmark here, that is actually here is a small transparent canal running through the various I say running through this, now you can say from running through the vitreous and reaches up to the posterior surface of the lens. This is called the hyaloid canal. In fact, hyaloid canal starts where your central artery of retina finishes. So, where your central artery of retina finishes, hyaloid canal starts. This indicates that distally it is called the hyaloid canal, proximally it is called the central artery of retina. Means hyaloid canal actually, and because initially this was called hyaloid artery, we did not call it the central artery of retina initially. So, the proximal portion of the hyaloid artery will become central artery of retina and distal portion gets obliterated here, and when it gets obliterated it will not remain an artery, it will remain like a canal. That canal will be called as the hyaloid canal, and hyaloid canal starts from where? From the center of the optic disc and will reach up to the posterior central point of the lens here, and this posterior side of the lens, and here where this is actually is attached, there is a small circular opacity is there. That does not actually affect the transparency of the lens. Small circular opacity actually will be present here on the posterior lens capsule, which represents just the anterior attachment of the hyaloid canal here. That is called the Mittendorf point, or you can say this is called the Mittendorf dot. This is called the Mittendorf dot. This represents the anterior attachment of the hyaloid canal, and the hyaloid canal is nothing but the distal vestigial part of the hyaloid artery, or distal obliterated hyaloid artery, and the proximal intact hyaloid artery is today called central artery of retina. Now, after the hyaloid canal, we have told you the vitreous is in fact is present here. Vitreous is very important, is a transparent colorless gelatinous mass that is actually present like a body. If it is like a body, it does not mean that it actually can be separated here. It is attached to multiple spots, like it is attached here to where there is this ora serrata, it is actually fixed with this. It is attached on the back of the lens also, capsule as well, there is some points here. It is attached here to the optic disc sides. So, it is actually attached like this, and it is very important. I have told you that it does not grow if it is damaged here. On removal, you say once removed, the vitreous will not regrow, it will not regenerate at all. It is a mass that has been actually is the mass of the proteins here. So, vitreous degeneration will lead here to the shrinkage, and shrinkage will actually cause here the globular shape of the eyeball will be surely will be affected here. It will be now smaller in size. So, the rays in fact are the focus, the object will not have its image on the retina, it will be beyond the retina. Beyond the retina means now you have to actually to focus this object or to focus this image, the object has to be held a little away from the eyeball, you see, away from your eyeball. You call this is the vitreous here. So, vitreous is important from this point of view. So, these are certain important areas which you have seen now. Now, especially when you come to this, after the vitreous and all these things here, if you will actually look at this now, for example, we are bringing a small piece of the section of the retina from here. So, we have here a retina now. Means if the retina is like this, so this portion I say it will be its 10th layer, and here is the vitreous, then will be its 9th layer, 8, 7, 6, 5, 4, 3, 2, and it will be its first layer now here. So, these are the 10 layers here. So, innermost will be the 10th and the outermost will be the first, and first will be now it is coming from your outer layer of the retina, that is the retinal pigmental as a pigment epithelium, and inner sensory layer will give rise to the 9 next layers, means from 2 to 10. And here is the vitreous, and here will be obviously the choroid, and here are the choriocapillaries. So, this is actually the section which we are cutting here and we are putting here on the board to show you how the retina looks like and how its cells are actually arranged and how these layers are in fact present here. This will be in our next session when we are bringing it various layers of the retina, how they are arranged and what they are, and from where in fact our optic nerve will generate, and where are our rods and cones, what will be in fact the spot where you are actually looking at these things here, and how these layers in fact they are arranged. We will actually see them now. Here in this diagram we are seeing the 10 layers of the retina in such a way that this will be actually its outer aspect from where you are actually looking at this. This is called a sclera, means this from outside we are going. This is a sclera. After the sclera, the next layer that will be actually is the choroid. So, we are watching here choroid. And then after the choroid will come the retina. And the retina means when you are actually coming from outside, in the first layer you will strike, and that will be called the retinal. It's called the retinal pigment epithelium. It's called the retinal pigment epithelium, which we are looking at. This is a single cuboidal shaped cells with the folded here. You see, and say as a membrane. You see that towards the retina to actually be having foldings here, and this is not firmly. You see that here from all the other layers of retina, means from 2 to 10 layers will be there. Towards the vitreous you are actually going here, till the last layer, 10th layer will actually separate it from the vitreous. That means it is actually sandwiched between the vitreous inside and the choroid outside. And it's you know that, and these layers which I am actually showing you here, it's called the layer number 2 onward up to layer number 10. This is now retinal pigment epithelium, and this epithelium is in fact in such a way that this is not firmly adherent with the next layers of the retina here. It is not firmly adherent with that, but outside with the choroid it is firmly adherent. The reason is that because it is getting its nutrition from the choriocapillaries, and in such a way that it is also having a tight junctions also. And due to these tight junctions, outside here the blood vessels, that is the blood component, will be separated from the retinal component, that is called the blood retinal barrier. That means your retinal pigment epithelium is doing this work also. At the same time, this is something that you are actually watching, and the retinal cells, they are very, very important in this sense. So much so that the outer segments of your rods and cones, you see here red color cones have been shown here, blue color rods have been shown, and the rods and cones, their outer segments which you are actually seeing. So, this is what we call here the first layer, it is called retinal pigment epithelium. Second layer, you are seeing the second layer, that is called a photoreceptor layer. It's a PRL, this is a photoreceptor layer, this is second layer. But if you are looking at this retinal pigment epithelium, this one which you are actually seeing here, there is a lot of functions you see, and rods and cones, their outer segments are embedded partially into the retinal pigment RPE, this is called the retinal pigmented epithelium. And at the same time, you see the rods and cones, we have already told you, in the periphery, the rods, periphery of the retina, it contains the rods more than the cones less. But in the fovea centralis, there was just the cones, no rods, rodless area. When you come outside here, the cones are and rods almost, they are almost equal, but in the periphery almost you have got just the rods here actually there. The quantity as concentration will be more here. And the cones, you can imagine that here, this all is outer segments of these epithelial cells, say outer segments of these rods and cones, they are embedded in the in the pigment, in the pigment epithelium here. Then we have got the inner segment also. So outer segment, then inner segment, then ellipsoid part, and then you have got the synaptic terminal. And the synaptic terminal, in case of rods, if you are seeing, they are called spherule, but if in case of cones, you say that in cones you call them the pedicles. But you see here, this is now the rods and cones layer, but after the rods and cones, you see this is the middle portion, the inner segment, in which you are actually watching here the cell bodies of these rods and cones. That is why this layer, that is actually called the third layer, as you can say. Even before that, if you see this is here, so you say rods and cones are there, and after the rods and cones you will see another layer. But before knowing this layer, you have to actually watch and have a knowledge about these cells. We have a lot of cells here. We have got this red color, we have got green, here red again, here red again. We have got a lot of cells, 4 different types of cells are there, but the 5th type of a very large cell is present here, that is called the Muller cells. Muller cells, they are present, in fact, you can say their cell body is lying in the inner layers, you can say 6th or 7th layer it is. So it will be around that, and it is giving its processes which will in fact will go here towards the initial layers, where they will spread and form here a membrane. And that is called the limiting membrane. And that limiting membrane is also maintaining, to some extent, it is also maintaining the blood retinal barrier also. In addition to the retinal pigmented cells tight junctions, this is another contribution for the blood retinal barrier, outer blood retinal barrier, that is called. But if you are actually looking at this, this is the third layer. This third layer is called outer limiting membrane, OLM. So this is formed by the short processes of the Muller cells here. Not only this, but after that will come here the now that area of that zone, or you say portion of your rods and cones which contain the cell bodies, which contain the nucleus. That is why that is called the nuclear layer. We call it from outside here. That is why this layer is called 4th layer. You call it now outer nuclear layer. So outer nuclear layer is nothing but the layer containing the cell bodies of the rods and cones. Now after this, you see that is coming here nuclear layer, then is coming here the synaptic terminals of the rods and cones, and now this is the time the synaptic terminal means the stimuli that have in fact have entered here from the 10th layer, has travelled through all the layers, have in fact gone into the pigmented epithelium. So light has been actually absorbed by them, and whatever light photons are there, they will be remaining absorbed here by the choroid, which is overlying, that is black also, and whatever photons actually return, they will strike the outer segments of your rods and cones, where the photopigment will be actually bleached, and here action potential is generated. Means action potential means the transduction will take place, and if the transduction takes place here, then this transduction of these impulses in fact will be handed over by the rods and cones to the next order of neuron, first order neuron. This is something receptors actually hand over their signals to the first order neuron, peripheral axons of the first order neuron, whose cell body is lying in the dorsal root ganglia or the sensory root ganglia. Same here, their analog, their homologous structure just equal to that will be actually lying here, these types of cells. You see that every first order neuron lying in the central, lying in the dorsal root ganglia or sensory root ganglia or any other ganglia, they are pseudo-unipolar. All are pseudo-unipolar. All general senses first order neurons are pseudo-unipolar. Even in special senses, test is also pseudo-unipolar, but remaining all special senses, they have got the bipolar. So here comes the first example of the bipolar first order neurons which are lying within the retina. These are the bipolar cells. You can see these bipolar cells. And these bipolar cells, they have got actually the dendrites which are synapsing with the synaptic terminal of the rods and cones. And I told you that in case of cones, especially those who are located within the fovea centralis, there is 1:1 ratio, one cone synapsing with one bipolar neuron. And after the bipolar neuron, when you are actually looking at here, so this is the spot where your rods and cones are the photoreceptors. They are transmitting their signals, impulses into the bipolar cells here. That area is in fact right in this area. You will actually be watching that there are synapses here, And in these areas, we have the dendrites of the bipolar cells and the synaptic terminals of the rods and cones. So, to regulate that area, to in fact modify this impulse transmission from the rods and cones to the bipolars, here another type of cells are actually located, which in fact will regulate this type of a transmission present here in this area. These cells are called the horizontal cells. And these horizontal cells in fact will join all these terminals, means the terminals of the rods and cones with the dendrites of the bipolar cells, are joining them in such a way that here a kind of a plexus will be created here. And that plexus is called the plexiform layer, and you call this plexiform layer is the layer number 5, and you call this plexiform layer as the outer plexiform layer. So, fifth layer is outer plexiform layer. What is this? Outer plexiform layer is a plexus like appearance here, the plexus like fibers here, which in fact are used, are say literally extensions of the horizontal cells fibers here, which in fact are making here a kind of a plexus at a spot, and they operate at an area where there is a joining between the synaptic terminals of the rods and cones, or the receptors or the photoreceptors, with the dendrites of the bipolar cells here. To modify them, we need actually a layer here, that is called the outer plexiform layer. And this outer plexiform layer then will come surely, because the dendrites are finished here, and now you are actually bringing here the cell bodies of the bipolar cells, and here this time this is called the nuclear layer, because there are the cell bodies of the nuclei of the bipolar cells are visible, though in this area you are watching actually the cell bodies of Muller cells also. So, this is actually called as, this one is called the bipolar. You call it the bipolar layer, you call this one as the nuclear layer, actually we call it the inner nuclear layer. The outer nuclear layer already you have seen, this is actually rods and cones cell bodies. I say rods and cones nuclei, but this is the inner nuclear layer, that is the nuclei of the bipolar cells, means bipolar cell bodies are lying there. But in this zone actually your Muller cell bodies are also lying there, having their short processes and long processes and multiple other processes also. So, here you are watching the inner layer here, and after that you see that are coming here the axons of the bipolar cells now. Axons of the bipolar cells, just like the cell bodies in the dorsal root ganglion, just in general senses here. There are central axons entering the central nervous system, and where is waiting? Second order neuron cell bodies, and here is again the waiting second order neuron cell bodies, that is called the ganglion cells. So, you call this layer as the ganglion cell layer. This is GCL, I will give this number also, but you see here this is called the ganglion cell layer, and this is basically 8th layer. Okay, this is 8th layer. So, where is the 7th? And now you have already seen here, there are the dendrites of the ganglion cells. This is the ganglion cell, second order neuron. Second order neuron is lying in the central nervous system. Normally, second order neuron is lying in the central nervous system. First order neuron is comprised of the nerves and nerve roots, etc. But second order neuron is always lying within the central nervous system. Why the second order neuron is lying in the retina? Actually, second order neuron is lying in the retina because retina itself is the gift of the brain to the eyeball here. That means it is actually is a part of the central nervous system. And all these neurons which we are actually showing you here, they basically come from the neuroepithelial cells here, except the Muller cells. The Muller cells, they actually develop from the neural crest. And if you are actually watching here, so inside we have actually macrophages, microglia, etc. also, they also develop from the mesenchyme. They are not also neuroepithelial, but this Muller cell is also not neuroepithelial cells. These are also neural crest cells. One of the derivatives of neural crest cells is Muller cells of the retina. And this also inner, and now this is the zone which you are actually watching here. Which zone? This is the zone where your bipolar cells, they are synapsing with the dendrites of the ganglion cells here. Right at this place, another, just like here, when there was an area when there was a synaptic terminals of your rods and cones were joining or synapsing with the dendrites of the bipolar. Here the ganglion, I say bipolar axons are joining with the dendrites of the ganglionic cells here. Right at this place also, it is not 1:1. It can be bipolar, one bipolar can actually multiple ganglionic cells will be approached by this one, but this whole area will in fact be connected with each other, and to modify this transmission process here, another type of small cells are required here. These cells are actually not macro cells, These are amacro cells, we call them amacrine cells. Macro means they are not macro, they are very small cells. We call these cells as the small cells. So we call these small cells, we call them amacrine cells. So you can see these cells, the red color, these are the amacrine cells here. They give their lateral projections, lateral fibers here and there, so that the plexus will be created here actually. And that plexus is in the area of the connection between the axons of the bipolar cells with the dendrites of the ganglionic cells. That is why this 7th layer is called inner plexiform layer. This is inner plexiform layer. Finally, the transmission has reached now the fibers. These are the axons of the second order neuron. Second order neurons, axons always actually ascend. If you consider retina as a part of central nervous system, so this type of say axons bundles should be called as the tract, because the second order neurons axons are called the tracts. Here we do not call the tracts, we call them optic nerve fibers, yet not optic nerve even. And here these fibers are okay, these are fibers okay, optic nerve fibers, this nerve should be myelinated, nerve is myelinated but not the fibers. As long as the fibers are in the retina, they are unmyelinated. But once they will leave now, the optic disc will come outside here, they are now myelinated. And these myelination of the nerves usually is by the Schwann cells, but this time is not Schwann cells myelination. Myelination is given here by the oligodendroglial cells here. That is why the multiple sclerosis, this is actually the autoimmune problem related here to the autoantibodies against the oligodendroglial cells. So this will be the only nerve that will be affected. Remaining all the nerves are actually saved, are they spared? Why? Because their myelin sheaths are actually developing from the Schwann cells. But here the myelin sheath is developing from the oligodendroglial cells. That is why the tracts are involved, nerve will be involved also this time. Tracts are inside, nerve is outside. Why it is involved? Reason is that because the myelin sheath is developing from oligodendroglial cells. That is why the multiple sclerosis can lead here to even blindness. So if you are actually seeing here the ganglion cells, and now all the fibers coming here, these are the axons of the second order neuron. We do not call them the tract, we call them the optic nerve fibers. This is actually called the nerve fiber layer. This is our 9th layer. Okay, this is the 9th layer, and all these layers I have told, I am actually telling you, they are all developing from the inner sensory part of inner sensory layer of the retina, of the optic vesicle or optic cup, you can say. If for example you are seeing that if all these fibers are now coming, you can expect that all these fibers can actually shoot up and will go in front here into the vitreous. But here Muller cells will again come into play, and they are actually stopping them, so that all these fibers from any ganglion cell will not trespass, will never cross and penetrate this membrane. This is the 10th layer, that is actually called the inner limiting membrane, is ILM. This is the 10th layer, which is being actually formed here by the long processes of the Muller cells here, as the Muller cells in fact will be giving here like this, so that all these fibers which in fact will come here will never come here into the vitreous, because here vitreous, it is a kind of you say is inner limiting membrane, it is a barrier between the vitreous and the retina, and here this is here. So all the fibers here, from all the fibers which I am actually showing you here, all nerve fibers, optic nerve fibers here, they will actually be leaving one spot here, and that one spot, that will be hardly 1.5 mm in the retina, that will be actually be made here through which all these fibers will rush now. A rush here, a disc will be created, that is called a rounded disc will be created, that will tell you here. Beyond that, its name now will become optic nerve. And this optic nerve, these fibers are there, so optic nerve will come from one side and from other side also through the optic canal, and immediately the fibers which come from the retinas, those fibers which come from the nasal side of retina, they will immediately cross. There is again second order neuron, they are coming from the ganglionic cell. Your optic nerve is basically the ganglionic cells axons bundles, and here the nasal fibers will go actually with the crossed fibers now. After that, the same second order neurons, this collection, and this area is called the optic chiasma, and this now it is called the optic tract. Now you will say it is optic tract, though it is second order neuron, even though optic nerve is second order neuron. But optic nerve, this is not called the tract. Otherwise, anatomically and functionally, this is a kind of a tract, but we will not call it the tract. And beyond that, even in the retina, we do not call it the nerve even, we call it the optic nerve fibers until it gets packed here and covered with the meninges, then you will actually be seeing this is coming back here like, and when the optic nerve is formed, you can say will have now, okay, it will have now the covering of the pyometer, and that covering of pyometer will go continuous over the covering of pyometer of the brain. That will have now covering of the arachnoid. That same arachnoid actually will become back here, will be actually surrounding the brain also, and the third will be the pyometer, I say it will be the dura mater. So that means our optic nerve is having 3 meninges here, but when the optic nerve actually reaches up to the back here, up to the lamina cribrosa, that is actually the plate of the sclera here, meninges stop there. Meninges will not surround the eyeball, they will become continuous with the various layers of eyeball, this is true, but they will not actually be surrounding the eyeball. Means we have a dura mater, but the dura mater will surround now and be a part of, I say it will become merged, or say become continuous with the first layer of your eyeball. Now you say it is sclera, means dura mater will become continuous with the sclera. Okay, then the next what will happen? Next we have got the arachnoid and we have got the pyometer, and in between both of them we have got subarachnoid space also. So what will happen with these 3 landmarks? Actually pyometer and arachnoid mater, they are actually a single layer. Here they are separate to give you a space, this is called subarachnoid space, but basically it is a single layer. That single layer, everywhere it is true, between that is the subarachnoid space, but once it will actually be a part of the eyeball, they will become fused here. There is a single layer, and that single layer will become continuous with the choroid. So if it is becoming continuous with the choroid, that indicates, that means it is becoming single layer. Before the optic disc or before the area where from the optic nerve starts, it was a double layer. It was having outer arachnoid and inner was the pyometer, and between there is a subarachnoid space. Means when the layer becomes single, this indicates that the subarachnoid space will be no more. Means around the eyeball we don't have any subarachnoid space. Means in the choroid we don't have any subarachnoid space, because it is becoming single to become continuous with the choroid. Means your subarachnoid space stops here at the back of lamina cribrosa. That's why if the subarachnoid space has got the more pressure, intracranial pressure, more pressure will be over the lamina cribrosa, and pressure is over lamina cribrosa, means this pressure will be actually reflected on the optic disc, that will actually bulge here. Intracranial pressure raised through ophthalmoscope, you can actually see in the fundoscopy the intracranial raised pressure here by seeing the bulged optic disc here. And after that, okay, then is the optic nerve fibers are there. This time you call them optic nerve, inside it will be optic nerve fibers, means it will become continuous with the retina here. So retina's various layers in fact will happen like this here, and their functions you will in fact be knowing more in detail in your slideshow. Let's watch the slideshow.